starting in 2 minutes. We are going on live.
Okay.
Another 4 minutes, we will wait.
Yeah, okay, sir.
So who is the director of the dryland CREDA now?
Just now, Dr. V.K. Singh has joined.
Who was in IRI, the Division of Agronomy.
Division of Agronomy.
Okay. So he will take it over now soon.
Yeah, already has joined.
Or joined. And then you'll have someone else in IRI now. As the head of that.
It will take some time.
Some time, yes.
Yes, sir. I think we are on live now. Yes, we are on live.
Let us wait another 2 minutes. Just time is given at 10 o'clock. 2 minutes. My time is 2 minutes lesser than.
Actually, we are on Zoom, sir. The participants will be on YouTube, so it will be broadcasted 1 minute late to them. That is the reason.
Okay.
No problem. We'll be starting at 10.
Fine.
Yes, sir. It is a time. So, Abhigna, could you please start?
Yeah. Good morning, good afternoon, good evening, distinguished participants, ladies and gentlemen. I'm pleased to welcome you to the webinar on breeding for efficient root system for water and nutrient uptake in plants. In this virtual conference room, I see such amazing guests and dignitaries from around the world sharing same interests and enlightening one another with the advancements in the field of agriculture. I am sure we are also reaching out to many new practitioners, technologists, innovators, business professionals, and civil society representatives, fellow mates, and many more people. A warm welcome to you all. We all understand the importance of science, technology, and innovation in our day to day lives and the ways in which they are transforming the world. Equally important are deployment of scientific inventions, innovations, and technological solutions on a scale that will reach the target audience. And hence, we are gathered here today on our Plantgenomia platform to obtain knowledge on how crop plant survival and fitness are dependent on root system architecture. Why here? Why today? What is the purpose? We are all embraced by the presence of the distinguished and well rounded dignitaries to shed some light upon the varied measures to be taken under consideration for the breeding strategies. All our online webinars aim to discuss leading edge technologies and recent scientific developments in addition to the immediate challenges in agriculture and allied sectors. The idea is to familiarize students and professionals about current research trends, relevant and pioneering technologies in plant genomics for high-level analysis and interdisciplinary areas that are embraced in leading laboratories by the excelling scientists. Plantgenomia. It is a non-profit organization promoting and publicizing genome sciences and investigations in the field of agriculture across the globe. We organize various webinars and developments comprehending genome research in plants for all the students, researchers, industrialists, and professors internationally. Here at Plantgenomia, we hold a global team of reputed scientists and research scholars from various parts of the world, distinct states of India, the United States of America, United Kingdom, Nigeria, Philippines, and Spain, etc. Technical support is laid to us by the scholars from the Department of Genetics and Plant Breeding. Our motto is to disseminate every bit of knowledge among all. We believe that no research is accomplished until broadcasted, and that it is an ongoing process through layers and lapses of time. I would now like to extend my heartiest welcome to the senior dignitaries, respected chief guest, Dr. C.H. Srinivasa Rao, Director, ICAR-NAARM, National Academy of Agriculture Research and Management, Hyderabad, India. Our speaker today, Professor Kadambot H.M. Siddique, Hackett Professor of Agriculture, Chair and Director of the UWA Institute of Agriculture, the University of Western Australia. The course subject specialist team, the course support team, our esteemed founder, Mr. Alamuru Krishna Chaitanya, and the lovely audience. I would then like to introduce Dr. Rupali, who had graduated with PhD from the University of Western Australia and presently working at the Department of Agriculture and Food, Western Australia, to introduce our revered chief guest, Dr. C.H. Srinivasa Rao.
Thank you, Abhigna, and thank you everyone involved in Plantgenomia for giving me the opportunity to introduce our honorable chief guest for the event, Dr. Srinivasa Rao. Dr. Srinivasa has a PhD in Soil Science and Agricultural Chemistry from Indian Agricultural Research Institute, New Delhi, and postdoctoral degree from Tel Aviv University, Israel. He is a fellow of National Academy of Agricultural Sciences, Indian Society of Soil Science, Indian Society of Pulses Research and Development, and Telangana Academy of Sciences. He is presently a director at ICAR-National Academy of Agriculture Research Management, Hyderabad. He had previously served as a director for ICAR Central Research Institute for Dryland Agriculture, Hyderabad, India. He also served as a project coordinator for All India Coordinated Research Project on Dryland Agriculture, Hyderabad, India. His areas of interest are climate change, contingency planning, soil carbon sequestration, rainwater management, climate resilient villages, rain-fed mission development, climate and conservation policy, agriculture research management. He has innumerable long-standing national and international networks and collaboration. He is a board member for National Council for Cooperative Training, Ministry of Agriculture and Farmers Welfare, Government of India. Committee member of Agriculture Research Service, Indian International Dryland Development Commission, Egypt, Asian Long-Term Experimental Network for Agriculture, Japan. He is a president of Association for Management of Agriculture Research and Agripreneurship, Hyderabad. Also a president of Association for Innovation Development of Entrepreneurship in Agriculture. He is an elected fellow for the National Academy of Agricultural Sciences. He has won many awards and recognition for his outstanding contribution to agriculture science and management. To name a few, Rafi Ahmed Kidwai Award, Lifetime Achievement Award, Golden Jubilee Young Scientist Award, ICRISAT Millennium Science Award, Vasantrao Naik Award for ICAR, Padma Shri Dr. Subbarao Memorial Award, Sardar Patel Outstanding ICAR Research Institute Award. He is chairman of Standing Committee for National Mission for Sustainable Agriculture, which is one of the 8 missions under Prime Minister during 2014 to 2017. He has also won Government of India Award for Best Emerging Technology Business Incubator, and the list goes on. Dr. Srinivasa Rao is a highly prolific author. He has published about 277 research articles in national and international journals. He has co-authored 26 books and published 10 policy papers on agricultural management practices. Dr. Srinivasa Rao Thank you for accepting our request to be a chief guest for our international webinar on breeding for efficient root system for water and nutrient uptake in crops. It is our extreme pleasure to have you today, sir. I now request you to take over the webinar session and speak a few words about your research in agricultural science and management. So over to you, sir.
Thank you, Rupali, and also briefing about me, and also other colleagues here, all your teammates, particularly a friend who welcomed me that from Plantgenomia, and all other your team members. Thank you very much for making me part of this meeting. And basically, topic is very related to my subject. In fact, recently we both, Dr. Professor Siddique and myself, we are making presentations on World Pulses Day at Indian Institute of Pulses Research, Kanpur, and I was also earlier working at IIPR Kanpur. He is a very eminent, basically we see, especially as my long association goes, I see is a pulse scientist, but I will introduce details of his expertise. Basically, I'm soil scientist and climate scientist, and also Alamuru invited me for this. You have to be here today, Professor Siddique. Yesterday, we thought that I'll be uncertain that I'll be arriving back to Delhi, from Delhi to Hyderabad, but he told that somehow he made me to be a part of this meeting. Alamuru, thank you very much. And actually speaking, my current interest is climate policy and agriculture policy development. Earlier, as you said, Rupali, I was working at CREDA in different capacities as director and project coordinator. And one of the, basically, I was working on soil carbon sequestration and climate change. And what we have done was at CREDA, we have developed district agriculture contingency plans for 650 districts. For example, climate change is a very important aspect, and overall rainfall has not decreased in India, but number of rainy events have decreased. For example, in Hyderabad, we used to get on an average one rainy event every week, but out of 700, 800 mm. But last year, just 4 rainy events happened, and it was, so there was a lot of mid-season droughts, which is similar to Australian conditions. So we developed 650 agriculture contingency plans towards climate preparedness, weather preparedness. So these 650 agriculture contingency plans were implemented across the country based on weather forecast by Indian Meteorological Department. We used to sensitize all the stakeholders, district officials, principal secretaries, and seed companies, input companies, so that if rainfall is less, rainfall is more, heat wave, cold wave, floods, seawater, etc., so that in 140 million hectares in India, the sowing area reduction is not reduced, and our country's food security is stable. This year, 295 million tons of food grain. We also have helped to SAARC countries how to combat that climate preparedness in similar way. And another work, my interest is climate resilient villages. So we wanted to see carbon positive villages across India. We have developed carbon positive villages in 151 district model villages in 151 districts, but state governments are implementing large scale replicating. For example, government of Maharashtra is replicating 5,000 villages by almost 4,500 crores, so that those villages with the community involvement, how that food production and food systems are stable. As a director of NAARM, we organize a lot of trainings from all young scientists. It is a great opportunity. I think Professor Siddique, I will be taking, this is opportunity. I would like to invite you, next batch of foundation course. All ICR scientists join at NAARM first. Then after 3 months of foundation course, they are posted in 117 ICR Institute. I am seeing many youngsters here, and whoever completes the ARS exam, so they will be cleared and get ARS, they will be joining at my institute academy, agriculture academy. So at present, recently we completed 111th foundation course. So all 6,000 scientists of ICR completed that foundation course at NAARM. For example, I belong to 40th foundation. So recently 135 scientists have completed training, and they are in the different institutes. And also not only young scientists, when they join as a directors, they come back again for training. When they join head of the departments, they come back. All 700 Krishi Vijnana Kendras, KVK coordinators, they will come. All project coordinators, they will come to NAARM. And we also recently started student entrepreneurship. We have reached to 25 universities. How to promote? We want to see agriculture exports, agriculture value addition. So at present, Indian exports are 31 billion US dollars exports. Government aims to increase to 62 billion US dollars. So we are promoting a lot of agri entrepreneurship, agri innovation, agri startups. So this is and also we train Indian forest officers from Indira Gandhi Forest Academy, civil servants, Indian IAS people from Lal Bahadur Shastri Academy, and also we go to Police Academy. So among 4 academies, Agriculture Academy is one. So this is my as you suggested, it is my brief background, but now NAARM is emerged as a think tank institute for ICR, just like Niti Aayog to Government of India. And thank you very much, Professor Siddique. Very pleased to see you always, and with your scintillating and lectures. And today we often meet in scientific forums, but today I'm very happy. These youngsters are grouped together, and youngsters under the name of Plantgenomia, they formed as a platform, and inviting eminent scientists like Professor Siddique. Earlier also I was invited, but I could not join. Alamuru earlier was also asking me, but I was again out of station. But today I am very, very pleased to welcome Professor Siddique. I have worked at Tel Aviv University, Israel, on root morphology. And in drylands, roots are very, very important. And when roots are so important, what are the efficient ways for breeding? Very, very important. But at the same time, when root systems are there in different crops, particularly if we talk different crops, we have studied in Tel Aviv University, we have largest aeroponic laboratory in Israel, Tel Aviv University. We have quantified, in fact, for optimum plant growth, how much root biomass is required. Within the root biomass, what are the components morphology is required, whether lateral roots or taproot or other parts. Why we wanted to study about root system there was, In typical field conditions, when intercultural operations are done, some part of roots are damaged. If some part of roots are damaged, really that part reduced root biomass can really hamper the harvest index or grain. That was the interest what we have done at Tel Aviv University. And today I am very happy to see that Professor Siddique is speaking on the topic breeding for efficient root system for water and nutrient uptake in crops. Under natural resource management, Professor Siddique and India and Australia have similar conditions, and maybe tomorrow or day after tomorrow, food may not be a problem in India, but water is the problem. So efficient water systems are very, very important. And fertilizer use efficiency is drastically reduced. Just 50 years back, each kilo of fertilizer used to give 17 kilos of grain. Now it has reduced to 3.5 to 4 kilos. We are putting more and more fertilizer to get this food security. So both water and nutrients are very, very precious, and we are also spending a lot of amount on fertilizer subsidy in India. So in this background, your topic, your paper, your topic is very, very relevant. And thank you very much on behalf of Plantgenomia and also Alamuru and all young colleagues. Very, very pleased. I'm very, very happy to join with the youngsters. And I take this opportunity to introduce Professor Siddique. Professor K. Siddique has over 30 years experience in agriculture research, teaching and management in both Australia and overseas. He has developed a national and international reputation in agriculture science, especially in the fields of crop physiology, production agronomy, farming systems, genetic resources, breeding research in cereal, grain, and legumes, particularly pasture legumes and oil seeds. Professor Siddique's personal research and others with whom he collaborates, Australia has become one of the major grain legume exporting nations in the world. His pioneering research in chickpea has contributed enormously to the Australian chickpea industry, which is currently valued at more than 600 million US dollars per annum. He was designated by United Nations FAO as a special ambassador for the International Year of Pulses 2016 and was also recipient of Orbe Memorial Award, member of the Order of Australia AM, Western Australian Year of the Award, and several awards from China and other countries. Especially, he is a highly cited researcher, and in 2020, 2019, in Agriculture Science, Web of Science, he has published more than 500 scientific papers and book chapters. His publication list includes 13 books and 64 book chapters, 480 reviewed scientific papers, 21 referred conference papers, more than 100 technical publications. He has also developed an extensive network of scientists within Australia and established a diverse range of overseas, several countries, including India. The key message, perhaps, all our youngsters would move forward from the many elements of Professor Siddique. He is a hardcore researcher, agriculture researcher, and good communicator, good writer, and very good science communicator. Perhaps the youngsters will take forward, having done research in India, particularly Indian students, I tell, they do very good work, but science communication is very, very insufficient. Perhaps as a role model, Professor Siddique, we all follow, and how to communicate to various stakeholders. Stakeholders in the means that stakeholders how to communicate same content to the farmer, same content to the district administrator, same content to other than your subject. Here is a genomics is a subject. If you want to explain about CRISPR-Cas technology to the extension scientist, how to communicate, to the other soil scientist, how to communicate. So science communication is an art. Perhaps we all learned from Professor Siddique how to communicate, having done good research. Thank you very much, Professor Siddique, for accepting invitation on behalf of Plantgenomia and also the core committee here, Alamuru, Rupali, and also Naria, and all I'm seeing Sujaya, Sri, Aman, and many more. Thank you very much. Now, floor is yours.
Thank you very much, sir, for apprising us with such sagacious knowledge and insight, for accepting our invitation and being a part of this event. It's been a stupendous commencement. Next, we have the much admired founder of Plantgenomia, Mr. Alamuru Krishna Chaitanya, to address the audience and guests present with us today, and to remark the initiative taken up by Plantgenomia and his team.
Thank you, Abhigna. Am I audible?
Yes.
Yes.
Yeah. Dear distinguished chief guest, Dr. C.H. Srinivasa Rao, Director, ICAR-National Academy of Agriculture Research and Management, NAARM, Hyderabad. Esteemed today's speaker, Professor Kadambot Siddique, Director of Institute of Agriculture, the University of Western Australia. Reverenced media partner, Just Agriculture, DPS Badwal. Scientists, professors, industrialists, senior executives, and all my colleagues. As we have such an enthusiastic audience from various parts of the globe, I wish all good morning, good afternoon, and good evening to everyone due to difference in the time zones. First and foremost, I'd like to express my heartfelt thanks for all for taking the time to attend this international webinar on breeding for efficient root system for water and nutrient uptake in crops organized by Plantgenomia. The way moving forward, together with us, we have distinguished guests and speakers who not only have a high command in authority of sharing their views, but also a living legacy in scientific research. Our inspired speaker, Professor Kadambot Siddique, will share with us on the valuable insights on breeding for efficient root system for water and nutrient uptake in crops. Global agriculture urgently needs crops with greater nutrient efficiency, meaning greater yield under suboptimal nutrient availability. The accelerating effects of global climate change are likely to accelerate soil degradation and limit the crop yields, as Srinivasa has mentioned. These facts have several important implications of crop breeding. Opportunities to breed more nutrient-efficient crops by improving root phenotypes is coming challenge and need to be done and sustain the future agriculture and the soil health as well. We are fortunate to have with us a great expert from that field to share the beautiful insights of the research in this area. Thus, facilitating everything, Plantgenomia has taken an initiative in meeting with all the world-class scientists to create a smarter dissemination virtual system by making use of the knowledge and advanced technologies, which benefits all the researchers to get maximum results. I was very happy to see the overwhelming response from participants from many countries touching almost every part of the globe. Once again, on behalf of Plantgenomia, I like to sincerely convey my thanks to all in making this event possible. And I do hope that you'll find the event more meaningful and as well as memorable. Thank you.
Thank you very much, Mr. Krishna. Those were some very gripping words coming from you. While we are presenting a gesture of gratitude, I would also like to thank Dr. K.P. Vishwanathan, Vice Chancellor of Mahatma Phule Krishi Vidyapith, Rahuri, and Dr. K.V. Peter, former Vice Chancellor of Kerala Agriculture University, for giving their valuable forward message on Plantgenomia webinar. Moving further, we would like to express our indebtedness to our chief guest, Dr. C.H. Srinivasa Rao, for his inestimable presence. Now, we subsequently proceed with the events. Before going to the estimable talk, I would like to call upon Dr. Rupali for introducing our today's speaker, Professor Kadambot Siddique.
Thank you, Abhigna. So on behalf of Plantgenomia, it is my greatest pleasure to introduce our speaker, Professor Kadambot Siddique. He has a PhD in environmental and crop physiology from University of Western Australia. He is presently an acting chair and director for University of Western Australia Institute of Agriculture. Professor Kadambot Siddique has over 30 years experience in agricultural research, teaching and management in both Australia and overseas. He has developed a national and international reputation in agricultural science, especially in the fields of crop physiology, production, agronomy, farming systems, genetic resources, breeding research in cereal, grain and pasture legumes, and oilseed crops. Professor Siddique's publications are considered as key papers in the above fields and are widely cited. These publications include numerous highly cited papers on adaptation, physiology, and genetics of crops to dryland environments. Professor Siddique has conducted research on adaptation of crops to water deficits and the phenological, morphological, physiological, biochemical, and genetic traits that enable crops to cope with various abiotic stresses. As a result of Professor Siddique's personal research and with others with whom he collaborates, Australia has become one of the major grain legume exporting nations in the world. His pioneering research on chickpea has contributed enormously to the Australian chickpea industry, which is currently valued at more than $6,000 million per annum. He is a fellow of the Australian Academy of Technological Sciences and Engineering, African Academy of Sciences, Australian Agricultural Institute, and Indian National Academy of Agricultural Sciences. Professor Siddique was designated by United Nations Food and Agriculture Organization as a special ambassador for the International Year of Pulses 2016. He has developed an extensive network of scientists within Australia and also established a diverse range of overseas, including China, India, Turkey, Syria, Iraq, Iran, Saudi Arabia, Oman, Malaysia, East Timor, Nepal, Bangladesh, Pakistan, Europe, Canada, and USA. Collaborative research and educational projects. Professor Siddique is the recipient of national and international awards, including Urbe Memorial Award, member of the Order of Australia, Western Australian Year of the Award, the Danhunag Award by China's Gansu Provincial Government, and the Friendship Award from the Chinese Central Government, which is the highest award for a foreign expert, in recognition of his outstanding contribution to agricultural science and education in China over the years. Professor Siddique is the highly cited researcher for 2 consecutive years, 2019 and 2020, in agriculture science, according to the Web of Science. He has published more than 500 scientific papers and book chapters. His publication list includes 13 books and special issue, 64 book chapters, 480 reviewed scientific papers, 21 referred conference papers, more than 100 technical publications, and 2 theses. His research papers are widely cited, particularly those on adaptation, physiology, and genetics of crops to dryland environments. He is a highly cited researcher in the field of agriculture science. Professor Siddique is on the editorial board of a number of international scientific journals. He has also trained numerous honors, masters, and PhD students. So Professor Siddique, thank you for accepting our request to be the distinguished speaker for today's webinar on breeding for efficient root systems for water and nutrient uptake in crop. It is our extreme pleasure to have you today, sir. I now request to share your screen to our audiences and proceed with the talk. Thank you, Professor, and over to you.
Thank you, Rupali, and I'll share the screen in a minute. And it is also pleasing to get an introduction from my own PhD student, Rupali, who completed PhD from the University of Western Australia. I also very happy to see Dr. Srinivasa Rao, who also spoke quite a bit about my experience, and of course we know each other for a long time as well. And I first of all want to congratulate the Plantgenomia for the initiatives. I remember Rupali and one of the other students contacted me and said, you are joining in this initiative, and I said, I think over the email, yes, please go ahead, and so on. So it's a very interesting concept, particularly from young people, particularly from students. So I would like to congratulate Alamuru, then of course Abhigna, Rupali, Sujaya, and all, plus also the senior members like Dr. Vishwanathan and KV Peter, whom I know behind the thing. I think this is shaping up very well. I looked at the list of people who have spoken before. So you have been able to get people, particularly during this COVID era, COVID time, and get knowledgeable people and get those wisdom to young people, because the future depends upon young people like yourself. And if you do very well, then the India and also the globe will be a better place to live. So with that, now I'm going to share my screen. So again, once again, thanks for inviting me for this. Is it clear now, or is there any problem, Alamuru?
Yes, sir, it is clear.
It's clear. So as you all know that I'm going to talk about an efficient root system for water and nutrient uptake in crops. Although I put breeding as a title, I will also refer to couple of agronomic practices. So the research is coming from various PhD students, and number of my postdoctoral fellows have done over the years. At the University of Western Australia, we have a very good group at the moment focusing below ground. This is quite important. Now, can you see the second slide?
Yes.
Yeah. So the outline of my presentation is, I'll give an introduction very briefly. Dr. Srinivasa mentioned about the climate change and abiotic stress, importance of crop root system, what are the opportunities we have. So once we know that we need to search for efficient root system, what is the efficient root system for one region may not be the same for another region. So it depends upon the environment, soil type, and the need of the day. So we need to phenotype a large number of populations. Then we also need to make sure that there is a genotypic variation if you want to breed. But then there is phenotypic plasticity, whether it is the root system or the above ground, so we need to understand that. What are the foraging strategies for the roots to take nutrients and water? Then let us look at how this is being used in the breeding program. Then I will draw some conclusions. I just want to say that whenever you do a talk, acknowledge first and finish it off. Some people, what they do, they put at the end. The problem is sometimes you run out of time, so then you don't acknowledge. So that's lesson number one I have learned over the years. So Australian Research Council through discovery projects, Grains Research and Development Corporation, which is largely funded by the farmers of Australia. Though, of course, my University of Western Australia, we also had funding from the CGIAR crop CRP grain legume program and several other programs. So this is the University of Western Australia. Those who haven't been there, this is a beautiful campus. And we don't have in Australia agricultural universities. When I first came to Australia, I was just amazed, why no agricultural universities? And soon I realized that that is best for any country, because in a comprehensive university like my university, we have medicine, engineering, mathematics, computer science, arts, humanities, etc. So agriculture is one component, so we can interact and integrate with other faculties. So we don't have, and I'm very happy. The university is situated on the coast, the Swan River, beautiful area. My office and laboratories are around here, and this is the Vice Chancellery, where the Vice Chancellor sits there. So it's a beautiful campus, very close to Perth City and the Swan River you can see in the background. And if those who have been to United States, particularly Stanford University, some of these, the tiles of the roof looks like a Stanford University. And our university is now 108, 109 years old. So it's not as old as some of the universities in Oxford or Cambridge. Now, if you look at the state of Western Australia, this is the capital where the university is situated. State of Western Australia is huge. To reach from Perth to this end, it takes about 4.5 hours, 5 hours flight. If I want to go to Sydney, it takes about 5 hours, 5.5 hours flights, and there is a time difference of 3 hours and so on. The beauty about Perth is that it is the same time zone as China, almost similar time zone as Japan, Indonesia, and also similar time zone of India, only 2.5 hours different. The next important point is that the state of Western Australia is the second largest state in the world. The first one is somewhere in the Soviet Union. So we have only about 2.6 million people. Can you imagine that? So state of Kerala, where I was born, has got 35.5 million people. So you can see that 2.6 million is about 10% of the population of Australia. And so very thinly populated, and hence we don't have any huge issue or major issue with COVID. The other point is that the Western Australia is typically dry land. Dry land means entirely depending upon rainfall, and hence it has got some challenges and at the same time opportunities. Majority of the grain, about 45–50% of the grain in Australia is produced in Western Australia, and it is all exported. Couple of slides, if you permit me. UWA, University of Western Australia, has a global reputation, an internationally recognized university. In various rankings, we sit on the top 100, which is quite good for a small university of only 25,000 students. For example, Melbourne University will be about 38 to 40,000 students. Sydney will be even bigger, and Monash will be bigger. Some of the big things which we appreciate is a well-established industry partnership, meaning for engineers or agriculture scientists and medical doctors, we connected with the community, we connected the industry. For me, my majority of my research, including the research done by Rupali, we get industry money, farmers' money, and we have been pretty good in getting that money to do our research, and then we do translate that research to the farming community. The last point on this slide is the Group 8 University. We are part of the Group 8 universities. There are 40 universities, 40 universities in Australia, and Group 8 is the leading university where about 70% of the research is done, and they are the oldest universities in Australia. Now, the other point is Professor Barry Marshall, who is shown here in the picture. He got Nobel Prize in 2005. That is quite an important thing for a university. And he is the man who said that stomach ulcer is caused by Helicobacter pylori, not by stress. So no one believed him. So as a young medical researcher, he did his medicine there at UWA. He taken a cocktail of the bacteria and he developed the symptom of stomach ulcer. Then he treated himself with antibiotics and he proved. And that is the thing got him the Nobel Prize. He's the director of the Marshall Center for Infectious Disease Research and a professor of clinical microbiology. I had the privilege and opportunity to travel with him in many parts of the world on various occasions. And he also occasionally consulted with me on agriculture and food security matters. I talked to you about the ranking. Ranking is very important. One of the ranking I'm listed here is, you can go and Google this, academic ranking of world universities. This ranking is based on metrics, the number of papers you publish, the citation of the papers, the Nobel Prize or various academy fellowships you get. So it's not on survey like QS and time ranking. It is purely based on metrics. So overall, UWA is now pretty good, 85 in the world in 2020. One of our graduates got the field medal, which is the highest in mathematics. So that helped us to move from 99 to 85. Let us look at life and agricultural science. So that ranking had agriculture combined with life science for a period of time. We were sitting around 24, 25. And 2017, they started separating after we lobbied that agriculture science should be separated from life science, and bingo, we are now 14, 14, 18, and 17. Last year it was 17. It's a very, very competitive environment. For example, you have to compete with very large agricultural universities, land-grant universities in the US, and also Chinese universities. So we are doing well for a smaller group, and we are first in Australia in agriculture science the last number of years. Can you see still my slides? Hello?
Yeah, yes, sir, we are able to see that.
Very occasionally, I'll keep on asking.
Yeah.
So,
okay.
So Australian agriculture and climate change, whether it is Indian agriculture and climate change, the principle is the same. The climate changes will add substantial pressure to agriculture. There's no doubt about it. And Dr. Srinivasa Rao mentioned that. In the case of Australian agriculture, if you look at this panel, you can see 1990 base and 2030, which is not far away. We're talking about only another 9 years. And we can see that temperature is increasing, rainfall is declining, except up in the bit in the north, and evaporation is increasing. There's no doubt it's happening. And for your information, Australia is the driest human inhabited country in the world. And Australia never had any agriculture. 200 years ago, the Europeans came, and everything what you see, whether it is the wheat, the cow, the sheep, the fox, anything you see is all introduced and adapted to and bred to local conditions. So the Australian agriculture is facing abiotic stress, example, terminal drought, acidity linked to aluminum toxicity, I'll come back to that later, soil compaction, again, I will refer to that, salinity and alkalinity, boron toxicity, low soil fertility, very, very highly weathered ancient soils. So the drying and variable climate will decrease water availability, and it will also exacerbate soil-related issues. So now let us look at the importance of root system. The root system is fundamental to crop productivity. You all know that. Roots are not only anchoring the plants, but for water and nutrient uptake. So it's critical to maintaining structural and functional properties. We'll come back to that. And so what I'm saying is that we need greater attention, breeding programs or agronomy programs, especially in the rain-fed environments, about roots. And people are not doing that because it's so difficult to see, difficult to select. Here is data which I haven't updated. If you look at here on the x-axis is the decade every 10 years, and the y-axis is the root studies, percentage of the plant studies. So what simply saying is that even now we are only about 35% of the papers published in a 10-year time frame is devoted to roots. So majority of the studies are done on shoot, and people doesn't even think about roots and then drawing conclusions. So I'm just putting a plea that any new PhD students and others starting think about roots also, not only the shoots alone. Now, if you look at the root traits, contribute to enhance to water and nutrient uptake. And for example, you can have a deep root system, and you can have, for example, reduced root density near the surface. I'll show you some results from some of our work. You can have deeper layers, greater branching at depth, and also deep roots, which can take up a lot of nutrients and water, particularly in deep soils. If the soil is not deep, then this particular trait is not much useful. Increase the root hair length and density. This is important. Root hairs are the surface by which the nutrients and water is taken up. The xylem diameter. So if you go and dissect your root system, you can look at, for example, decreasing the radial resistance, and also increasing the xylem size decreases the axial resistance. So if you look at the root as a like a pipe, so the flow of that water depend upon the length the root is there, but also the diameter of that pipe, if you look at that. So now let us look at the opportunities. What are the opportunities for improved crop productivity by selecting for root traits which are beneficial for nutrient and water uptake in a particular environment? So we can look at root architecture, genotypic plasticity, then we can look at foraging strategies. But the problem is, as I mentioned at the beginning of my talk, accurate and efficient phenotyping is an issue. So in this picture, I have shown a semi-hydroponic system which we have developed, and it is published. I'll show the paper. This is a legume plant lupin, which is growing in a bin system where we can absolutely trace the root system. I'll show some slides. Now, we can look at the root and rhizosphere. Rhizosphere is the area surrounding the root in the soil. Different approaches. We can have laboratory, we can have greenhouse. Ultimately, both Dr. Rao and myself believe in validating that under field conditions. So if you look at the bottom part, you discover, you evaluate, you validate, and you utilize. So for example, the basic research, the agar, test tubes, etc. You can look at gel plates, etc. Biochemical and genomic and cellular anatomy. You can look at plant growth using mutant screening, plant development, seedling screening, early screening for root traits, provided that early seedling is correlated with the later plant development. Nutrient deficiency, genetic diversity screening, root anatomy, recombinant inbred lines, NILS, near isogenic lines, and natural populations and ecotypes. There is a lot of resources now available in the gene banks. And this is where the translational research defined abiotic stress. Then you move into applied research, seasonal root growth. You can look at for the 3-6 months. Plant molecular breeding, multi-location field, and then, of course, you can use augers and shovelomics using shovels and so on. And more recently, we have been moving towards GWAS, quantitative trait loci mapping, biomass producing estimates, etc., plant microbe interaction. This is quite important because it's not just the roots alone. The soil is surrounded by numerous microbes, bacteria, actinomycetes, fungi, rhizobia, beneficial, non-beneficial. Ultimately, I think we need to go for marker traits or QTLs so that we don't need to go and dig roots each time. The breeders will not do that. So that's where the concept, I like this figure very much because it really encapsulates the idea in one slide. Now, I talked to you about the semi-hydroponic system. So it is simply a rubbish bin, which is about 1 meter and has got a large capacity. We don't believe in fully hydroponics. So we have some water and an immersible pump here. And what we have is that the nutrients, etc. here, and that pump then pumps and slowly brings up. And these are the pouch, which are waterproof pouches, and they mimic the dark environment in the soil. So you germinate your seedlings and transplant into this pouch, and you can just hold those plants by clipping. And the beauty is that we can open the pouch at any time and have a look at what is going on. For example, if you look at here, semi-hydroponics bin system on lupins, which is a legume plant, you can look at the first order, the second order, the tap roots. You can go and look at the fresh samples of your root hairs. So these pouches are no constraint. So it gives the true genetic potential of that plant. Of course, in the soil, it is different. So we have to take it in the soil as well later on. So that paper, this is where our technique is developed. So you can get all the details of that in the paper published in Functional Plant Biology. Now let us look at some data. So we have about 2,000 what we call collections of lupins in Western Australia. Western Australia is the largest producer of narrow lupin, Lupinus angustifolius. And based on various criteria, we studied 112, which represents the maximum diversity available within the genetic sources. So you can see from anything from root length, first order branch, root surface volume, all the way to root dry mass at each depth and so on. The main point I want to show is that with that methodology, we were able to show significant variation available within the germplasm, whether it is the root length from 99 or 100 to 1,793. Let's take a dry matter, for example, 42.5 gram per plant to 1,094. So we were able to characterize, phenotype, large set of lupin genotypes using that technology, which gives us abundant information in order to take it further studies with soils or in the field. So this project was funded by Australian Discovery Project, Australian Research Council Discovery Project. Very, very competitive. The success rate is only 9%. So they were very happy with what we found. So let us look at one such data. So this is the 112 genotypes on the x-axis and the tap root length, just one trait. Then if you look at this 2 weeks after transplanting, or 2 weeks after transplanting, you can see significant variation already started. By the time 6 weeks, you can see this genotype is sitting here, as I said, 1,700, and this is sitting around 250. So significant variation we were able to establish through this process. It's not only that trait. If you look at the number of first order branches in the 112, so you can see that I just plotted some, and you can see the variation number of first order branches in one genotype is here, and the highest is sitting here. Now, one of the beauties that the biophysical modeling, the modeling approaches available will help us to model root system for different times. For example, you people some of you are sitting in Hyderabad. And if you know the Hyderabad soil conditions and water, etc., and then if you know certain parameters, we can model what type of roots are required in Hyderabad, rather than going and testing another thousands of germplasm and spending a lot of time and resources. So there are 6 models. One is root map model, sim root model, root type model, SPC SIS model, RSWMS, and root box model. One of our PhD students, Vanessa Donabin, she developed this modeling root soil interaction, and we published that in Plant and Soil. So the model, in the case of sim root model, the allocation of carbon within the plant drives the root growth. So the carbon is produced after the photosynthesis, and then carbon is partitioned between various shoot organs, but also it comes to the root because root doesn't do the photosynthesis. Root takes the water and nutrients, but root should also get the resources. And the root architectural development, it can track the dry matter allocation accumulation. The root map, which we use, root growth is driven by the feedback between resource demand and resource uptake at the individual root. So it's a biophysical model with a clear physiology and genetics and the fundamental plant growth attached to it. Hope you still can see me.
Yes, sir. It is clear.
Very good. Very good.
Yeah. Yes, please proceed, sir.
Yeah, thank you. Now let us look at root phenotyping and root modeling. So I already told you the semi-hydroponic system. I showed you the root depth, the branching, root density in depth through that variation. Then let us look at 4 genotypes we have selected: 85, 60, number 71, number 4. And the one with the plant is actually measured through the semi-hydroponic system. Then we used 2 models. The model number 1, the first one is a root map, and the second one is sim root model. What it shows is that we were able to simulate rooting, root branching at an early stage up to this level. This gives us confidence that together with the modeling approach, together with the phenotyping approach in this bin system, plus then we are able to take them into the soil situation. I'll show you some data. So then what we did was we simulating root system and P uptake in lupins. So for example, what we did was that total root length and P uptake on 2 genotypes with 3 pre-treatments: nil phosphorus, top dressed. Normally some farmers practice. Nowadays, many farmers are banding below 5 to 10 centimeters. Then we looked at 3 genotypes, 71, 85, and 4 genotypes, 4 and 60, and more scenarios. And I'll show you how that is going to happen. For example, so that's where the narrow banded phosphorus is sitting. Here is a root system which is not very much branching on the surface. The band is sitting here, so the plant clearly missed out because you apply the fertilizer, you plant the seed, and it did not come. As a result, it missed that phosphorus. Now let us look at another type here. This type is very interesting. So this has got a very good branching here, and it really gone into the phosphorus placed, and as a result, it was able to utilize the phosphorus and branching. This kind of modeling can be used to test certain hypotheses or also test genotypes at different environments. And that's also published. Again, almost what I'm showing here is all published by one of my postdocs, Yinglong Chen et al. Now I'm going to show this is done in the soil in large tubes. Total root length centimeter per plant, 3 genotypes, 16, 44, and 85, and well watered at 23 days after sowing. And you can see that 44 and 85, there is a significant difference. But as you move into 49 days after sowing, and water stress, we imposed water stress, you can see this genotype 44 under well watered and water stress behaved similar, very little plasticity, whereas genotype 85 showed significant plasticity. This is important, again, when you deal with the dryland situations. If there is water later on coming in season, or if there is deep water, you need to choose a genotype which is has a plasticity in order to take up that water and nutrients, rather than genotype which is less plastic. Of course, the foraging mineral nutrients is very important. Majority of the flowering plants have mycorrhizal symbiosis for foraging mineral nutrients, particularly phosphorus. Family Proteaceae plants and some lupin species develop cluster roots to adapt to low P. It's like a bottle brush. That bottle brush has got a huge surface area where they can go and take up the phosphorus after solubilizing. Unfortunately, the narrow leaf lupin, and the same applies to chickpea, do not form cluster roots, does not develop effective mycorrhiza. So how are they going to take up phosphorus, particularly when the phosphorus is bound. Later on, I'll talk about it in the soil clay particles. So they develop adaptive strategies, altering root architecture and increasing acid exudation. This is quite important. I'll show some data. So here is an example of a rhizosphere, means the area surrounding the roots and the soil. Exudation in narrow leaf loop in related 3 genotypes in response to varying phosphorus supply. So you have 16, 44, and 85, and you have 0 phosphorus, same genotype, low phosphorus 50 micromolar, and high phosphorus, which is 400. So you can see different acids are produced. These are anions, citrate, malate, and acetate. If you look at point number 1, major acid is citrate. They excrete, they solubilize the phosphate, which is bound in the soil particles, and make it available. Significant carboxylase exudation at low P, particularly low P. The plant is then giving the signal that phosphorus is very low and start excreting, exudating more citrate, and that will help. But let's say if you take that plant to a high phosphorus soil, then the plant, the genes will say, no need to exudate and waste that energy. There is plenty of phosphorus available. So these were all possible for us to study because we were able to phenotype in a system. From there we understood. Otherwise, if I give this to a PhD student and ask her or he to select a genotype from 100 or 200, what will they do? So this is the way one needs to work out. Look at the variation, you believe in that, then you take and test the hypothesis. And this is also published in Plant and Soil. Now the crop which is more familiar to you, the chickpea, which is also quite important for me. As you know that something like 20,000 accessions and lines are held at ICRISAT and ICARA, and about 300 represent the what we call the reference set, some people call co-collection and so on. We have fortunately 270 of them imported to Australia as part of a collaborative project I have been doing with ICRISAT over the last couple of decades, I would say 15 years. And we included 270, and that represented 29 countries. And we used our semi-hydroponic phenotyping system, which I have already described. Here is the system. It's in the bin system, in the glass house. And this is how beautifully the plants are already growing in the pouch. And look at that. ILWC245, which has got a different root system. Look at our ICCV 95311. And again, this paper is available in Journal of Experimental Botany. This has helped us to do wonderful studies on phosphorus use, and also I'm now doing quite a bit of work on drought. So from the same study, just to show you the taproot length, and you got the genotypes on the x-axis, and this is the Desi type, which you all know what is Desi chickpea. Huge variation from here to there. And we had a smaller set in the reference collector, only 53, 55 Kabulis. Again, in the Kabulis, we can see the difference. And what we call the P shape or intermediary between Desi and Tabuli, smaller number, but again, variation. So the take-home message here is that the semi-hydroponic system which we developed was able to identify and screen efficiently the mini core or the reference set beautifully, and we have about 35 traits we were able to measure. We moved on, and then large number of wheat collected in Australian germplasm, and from there I brought 270, and these 270 now phenotyped, and that's also published in BMC Plant Biology, and wonderful traits. Again, you can see. Couple of points here. The total root length, the centimeter per plant, you can see this genotype is sitting somewhere here, 550, and look at that, something about 3,500. Sometimes people say that, okay, if you have a smaller above ground, then you have a smaller root system. Yes, that may be true, but in this case, both are more or less similar. And look at the prolific formation of the roots, both the nodal and seminal root in the wheat system. Now, this study is now used by a number of PhD students, and more recently, one of the students from Bangladesh is looking at the QTLs using some of the nil and drill lines, including some of them developed by Rupali. So here is one of my PhD student, Victoria. She's from Chile. She's got her PhD and gone back. So she tested the hypothesis that, okay, we people did first it in the semi-hydroponic system, but we have rhizo boxes, 1 meter, 1.5 meter with the soil, with the glass panel, and she was able to look at the images, and these are true 3-dimensional stuff. So we got different cultivars, and the main message here is that what we found in the semi-hydroponic system, to a large extent, it was true. In other words, a genotype with a large root system we found in the semi-hydroponic system also held true in a soil system when we looked at in a rhizobox situation. And then she looked at the effect of early drought on different root system. We do get the farmers plant the crop and they get some rain, and then up to 1 month there is no rain. So we simulated that in larger PVC tubes with soil, and she was able to then look at the importance of the root system and the ability to combat drought. That work is published in Frontiers in Plant Science by Rupali. Here is an early work which I did way back when I was active in the field. This work was done in the field for 3 to 6 months in the field. And I have, to summarize this here, I have the root dry matter, just to focus on the graph A, root dry matter on the x-axis, shoot dry matter on the y-axis, and then you got a 1-to-1 line. Just for clarity, 3 genotypes, Culin, which was the most modern variety, Gamania medium era variety, purple straw, which is one of the earliest varieties in Australia. So what you can see, TS stands for, those who know about wheat, terminal spikelet, A stands for antithesis. So in the case of a modern variety, the partitioning of the resources, let's say dry matter or photosynthate, started very early on to the shoot, less towards the root. Whereas in an old variety like purple straw, majority of the dry resources went into the root system. So what breeders have done is that over the years of breeding, over the 10, 20, 30, 40, 50 years of breeding, they have reduced the root system. It is redundant. The root system, huge amount of investment, but that is not necessarily in a modern agriculture. And I'll show some excellent example. I won't talk about the graph B here, and that's a highly cited paper in Plant and Soil. One of the postdoctoral fellow from Iraq came and spent time in my lab. And here, what we are showing here is the cumulative root length and then days after sowing. These are all done in the rhizoboxes, if you like, the soil. So 1958 release is on the top and 2007 release, which is the most modern variety. So you can see they had a lot of dry matter length in the old variety. That's about 46.8 centimeters per day. In the case of the modern variety, less root length, less dry matter in the root, 26.3. So it's very clear that the breeders have unknowingly reduced the root system and then put most of the resources into the shoot and then increase the harvest index in order to give more grain yield. So you believe it or not, we were able to trace that using our technique here. These are actual from the soil. So Heron was released in 1958, Gladius in 2007. You can see the dense, so 1, 2, 3, 4 plants here and 4 plants here, exactly the same time planted. So that's the evidence to show that the root system has been modified large extent without any, oh, it's a black box approach. They didn't do that, but just ended up. Again, that's a nice paper in Plant and Soil. So if you look at the year of release here and then root length density, generally there has been a decline from early time onwards, which I saw way back in the 90s. And also the total root length is declined, total root biomass is declined, but the specific root length has not changed. Other studies we have shown us a number of seminal roots per plant. It's not only the nodal roots, but the seminal roots is important. The simple point I want to emphasize here is that there is significant variation available in the gene pool for that trait as well. Now, this is done by another visiting fellow, the young man from Indian Agriculture Research Institute, Vijay Punia. Vijay, I'm not sure whether he's in the audience. He came and visited me, and he had the Australian Endeavour Fellowship for 6 months, very well organized, and the rest of my team here. What we have done here, just very carefully, I'll go through this. This is at early vegetative stage. This is at almost grain filling stage. Then you must look at here the soil depth, 0 to 1 meter. These are our 1 meter rhizo boxes. Now we have 1.5 meter. These are the glass panels where we can trace. pH of the soil, have a look at it, 4.81 to 3.5. Very acidic soil. Not only that, aluminum toxicity is increasing 1.25 milligram per kg to 28.5. Very acid soils. The problem is that durum wheat, these are durum wheat, cannot tolerate acidity. So one of my colleagues from CSIRO, Manny Dilhasi, he has been moving genes by backcrossing the genes they obtained from Brazil. Brazil has got a lot of acid soils, and that's called the TAM8-1B gene. He put the TAM8-1B gene in one of the Australian variety, Jandaroy, and look at that, magic. Look at that, they were able to grow. Look at that, the root just stopped growing. Of course, you may ask why then the plants are looking the same. In this particular experiment, we did not, we provided sufficient water for both treatments and also nutrients. Then by maturity, you look at that, this is fully filled, the other one did not move. Because we provided water and nutrients, the yield was very similar. But there is a wonderful study at the moment being reviewed by Plant and Soil by another Chinese visitor, where what we did was we used the same approach. We used 1.5 meter tubes, soil similarly packed. Then after anthesis, we did not provide any water to the some treatment, and as a result, no water uptake by the one without the gene, and harvest index collapsed. Grain yield became something like 60–70% less. So it's a wonderful paper. Hopefully, it will be accepted. So again, by modifying the root system, in this case, acid tolerance, and not by GM technology, we were able to show that they can tolerate the subsoil. And some of these techniques, like the semi-hydroponic system, the rhizo boxes, the tubes, etc., are very helpful to do. And of course, we have also taken this into the field, and the field data also shows exactly the same results. So you still with me, all of you? Hello?
Yes, sir. Yes, sir.
Good. So moving on, then we did all the Australian barley genotypes. So you can see that another visitor came and did a wonderful work, Jidong Wang, and you can see the beautiful differences in root architecture here. Then we moved about 10 of the from the 189 to do further work in the tubes with the soil. These are semi-hydroponics. We have huge collaboration with one of the universities, Northwest Agriculture and Forestry University in China. And here, some of the PhD students have been working on corn, maize, and again, both semi-hydroponics, and then we take into the soil and larger variation we were able to do, and some of the phosphorus use work. This is just currently being done. One of my PhD students from Bangladesh, Mohammed Salim, is looking at soybean with a different drought tolerance, which I got from University of Missouri in the US, collaboration with Henry Nguyen. And you can see that he's already showing significant differences. These are large rhizo boxes, 1.5. And from there, he will select where he wants to study the interaction between phosphorus and drought. And of course, this study, prior to this, we did the study in the semi-hydroponic system. So we were able to then take the contrasting ones to test that in the soil. Another PhD student, Renu Sharda Devi, she's originally from Kerala. What she did was that we created this tube and cut into 2 pieces, then put a compartment and then put it back together. So in other words, water and nutrients from one part cannot go to the other part. And then we developed a technique where wheat seedlings were germinated and they were put here. Half the roots goes into one compartment, half course to the other. So there were four genotypes, 3119 rice tail, which is very drought tolerant, and Wild Catcham, a Western Australian variety, and another breeding line. So you can see this pot is split, but the water cannot move, well watered during anthesis and maturity. And here, W stands for watered, drought after flowering, DD stands for after flowering no. So this way we were able to understand the signal coming from the dry part of the root to the plant and so on. And that paper contrasting stomatal regulation and leaf abscisic acid concentration with genotypes when split root system were exposed to terminal drought. Again, those who are interested can look at that. But the point I want to say is that there are different techniques where we can then test those hypotheses and identify the importance of root system. Renu went on and did another wonderful study where this is a 1 meter PVC tubes with the soil, well watered. Then we have some paraffin wax which separates. So we were able to dry the top, then supply water below, bottom got wet, and then of course throughout water stress after flowering. And this helped us to look at the stomatal behavior under terminal drought, especially after post-anthesis. I'll show you only one data. So here you can see that FTSW stands for fraction of transpirable soil water. So that means very dry here, 0. 1 means well watered. So there are two genotypes. Dry tail is a relatively drought tolerant variety. IGW is more sensitive. So you can see that how the IGW3262 completely opened its stomata and it collapsed very rapidly as the drought came. Whereas in the case of dry tail, it was more conservative. It was able to sense the drought will come. So it was able to use water sparingly and had a grain filling. So again, this sort of studies came out of that experiment which I showed. I mentioned that agronomy is important. So sometimes you don't need genetic approach alone. Deep ripping, Dr. Srinivasa Rao knows about what is deep ripping, and we have compacted soils in many of the mechanized countries like Australia because of the continued use of large tractors, particularly before the conservation agriculture commenced. So deep ripping, here you can see the deep ripping helped the penetration, whereas the unripped area, and as a result, very poor yield, and so the farmers do practice deep ripping occasionally. But now with the conservation agriculture, no-till technology, this is less so. And that's also we published in field crop research. The same data to show that in the case of deep ripping with a black histogram, you can see it allowed the root system to grow deeper compared to the non-ripped. Just about last week, we published this paper. This is a wonderful study done by one of my former PhD students, Yan Fang. She's an assistant professor in Northwest Agriculture Forestry University. It's basically what we're looking at is that we don't need a large investment of root on the surface soil. When I say about 0 to 30 centimeters or 40 centimeters, if you start having large root system in a dryland agriculture, because when the drought comes in, that is using all the water from the top and there is not much going on the bottom. So we tested that in the field. It's a busy graph, but I will just digest this. So you have 2016, 2017 season, 2017, 2018 season. Then you have rain-fed, you have irrigated. What we have is the root length density, centimeter per centimeter cube. Then you have irrigated and rain-fed. So if you look at the top portion, 0.0 to 0.4 meters or 40 centimeters layer, you can see the root system of the modern variety, which in this case is CH58 and LH7 was less, both under irrigated and rain-fed conditions in 2 years. Whereas if you have, if you look at the 0.4 to 1 meter, you can see they maintained that root system well. So this way they were able to take up water and nutrients, water in particular from the deeper layers, and in that way better nitrogen use as well. So that paper is published in European Journal of Agronomy. To summarize that work, I'll just show you this diagrammatically. Hope you can see that. Large topsoil roots in the 0 to 40 centimeters depth, before anthesis soil water use, and after anthesis. Increase pre-anthesis dry matter, reduce post-anthesis nitrogen accumulation, reduce post-anthesis dry matter accumulation, and reduce grain nitrogen per unit root length, and reduced grain yield, reduced grain nitrogen concentration in a dry land system. As opposed to that, if you have a small topsoil root genotype, as I showed earlier, and they can have more increased post-anthesis dry matter, more grain yield, and also better nitrogen concentration. So studies done in the glasshouse, then testing that in the hypothesis. So, and it clearly shows that modification of root system for a particular environment in the lowest plateau has produced more yield and a better nitrogen use. Now I'm going to move to the phosphorus part of the talk. So you all know, many of you have studied soil science, soil P fixation leading to low P availability. Very quickly, cations present in the soil: aluminum cation, manganese cation, ferrous cation, calcium cation. So for example, phosphorus, phosphate, plus aluminum becomes aluminum phosphate, not available to the plant. Phosphate, particularly in acid soils, phosphate anion, and manganese, magnesium phosphate. So let us look at the phosphorus cycle. Fertilizer factory, the farmers incorporate fertilizers. Phosphate come from the rock phosphate. And then what happened? You go into the soluble form, the roots, legumes, and cereal crops take up. Crop residues are taken. Livestock export from the food system, and harvested grains goes in. Manure comes in, decomposition occurs, soluble P again taken up, some soil is blocked or locked up, retained, and some is going into the water and coastal system. So our job is to efficient uptake of phosphorus by the plants and also reduce the locking up in the soil. Otherwise, the farmers continue to apply a lot of phosphorus in India and Australia, and huge amount of phosphorus is in the soil and unavailable form, and also it's not sustainable. And of course, unlike ammoniacal nitrogen fertilizers, phosphate is still coming from the ground or rocks. So that's depleting rapidly. And India needs a lot of phosphorus, and India doesn't have a lot of phosphorus deposit, so it imports from places like Morocco. So what are the diverse below ground P acquisition strategies for plants? So we can look at root architecture, spatial configuration, individual plant, rooting depth, root branching. You can also look at the root morphology, root diameter I mentioned earlier, specific root length, which is very important, root hairs, lateral root density, root tissue density, etc. And if you look at the physiology, improved mineralization of the soil nutrients, including root respiration and release of root exudate. I mentioned that in the case of lupin, but I'll show some more results. And don't forget root biota, interaction involving direct interactions such as nutrient capture. So for example, mycorrhizal fungi, it is a very beneficial, and also rhizobia in the case of legumes. And the pathologist, the root pathologist, has to look at the interaction between the pathogens and roots. Here is an example of root exudate in different legume species. This is bird's foot Trefoili, Lotus corniculatus. Then this is a French Ceradella Ornithopus sativus, and of course your chickpea here. You can clearly see that in the case of chickpea, most of the root exudate, although not different with in this graph when you express as a dry matter basis. But if I express as the length basis, that is length per unit length, then you can see that chickpea is producing enormous amount of root exudates compared with the other two species. Some of our phosphorus deficiency work, which we have done with all the co-collection reference set, you can see the typical classical phosphorus deficiency coming. This is the work done by my research fellow, postdoctoral fellow, Jain Pang, and published in Plant and Soil. You can see what is the early symptoms of phosphorus deficiency. But if you look at that, we have from the same collection, no leaf senescence at all. So here is an example of P deficiency. This genotype was not able to take up phosphorus from low P soils as opposed to this. So significant genotypic variation. And again, a lot of this is available and published. One such data I wanted to show. So on the x-axis, physiological P use efficiency, and on the y-axis. On the y-axis here, P utilization efficiency. And then you got the Daisy, Kabuli, and P shape. These are from the co-collection, and you can see the tremendous variation available for both. So physiological P use efficiency is shoot dry weight per shoot P. P utilization efficiency is shoot dry weight by shoot P content. So very easy. So the take-home message is that the co-collection of chickpea, we were able to screen for phosphorus use efficiency, phosphorus acquisition efficiency. We were able to see significant variation in that. So the enhancing P uptake current cultivars are poor fertilizer P, particularly in the case of cereals, because a lot of that is locked up and it's not available. But we could breed for a new genotypes, new cultivars, where some of the upregulated movement of P into the plastids and later on available to the plant. Other losses, example, microbial immobilization, leaching in sandy soils also occurs. So we need to make this efficiency happening. And here is a summary diagram. So the summary diagram shows that develop an easily measurable above ground proxy, because it's so difficult to measure the P use efficiency for thousands of germplasm. And performance-based approach, that trait-based selection, develop a crop root traits to improve P acquisition and lower external critical P requirement by conventional and modern breeding. But we can also look at the management practice here, optimize root-root interaction, intercropping, very much practiced in India, rotation, intraspecific plant density, and of course the microbial fertilizers are available these days, and the optimization of the input. So basically efficient P uptake and optimal input, and you can have accuracy, quantify imaging, unravel process of root, for example, X-ray and CT scan, novel sequencing, better understanding of the acquisition traits, specific environmental conditions under low P and high P conditions, and integration of multidisciplinary knowledge, soil science, physiology, and genomics work. So we need to understand the trade-off among root functional traits between improved capture of P and other resources. The problem is that, yes, we went and measured 300 and so on, but how can we measure carboxylase, which is below ground process? Measuring carboxylase is often laborious, could find an easy. So we're talking about the rhizose sheath, the soil surrounding this area. How do we do that? In our case, we were able to go and destructively measure, but in a breeder, they can't. Fortunately, what we found that the malonite was the major organic anion in the case of chickpea, which was useful for mobilizing that phosphorus. The malonite amount in rhizose sheath was highly correlated with the leaf manganese concentration, the fully expanded topmost leaf. This is very encouraging, and this was published in New Phytology, where we were able to show that there's a good correlation. Bingo. So at least the breeders can go and take the leaf and do the manganese, or now we are working towards the molecular markers, and because chickpea genome sequencing for the 3000 we published with Rajiv Vashni et al. Last year in Nature Genetics. Now we are in the process of publishing the 400 we published. We are now in the process of publishing the 4000. So this kind of approach is now leading into genomic approach, and the proxy leaf manganese concentration is very, very promising. I won't go into the details why that, but it's available in the paper. I'm coming towards the end of my talk. So we can look at now efficient root system in chickpea for drought resistant. This work was done by Kashiwagi, a Japanese scientist located at Ikurisat, probably Dr. Srinivasa will know him. He's now back in Japan. What he showed here is that you got different root length density at different depths in the soil, and moderate drought and severe drought. So this is done in the field. So overall, there is a good correlation between seed yield and root length density under moderate, and that increased with severe drought and different depths also contributing. From there, they went, from there, what we did was we looked at about 10 genotypes in large containers and looked at the behavior of roots. So we started water stress here. So this is well watered, about 80% field capacity. And this is a genotype's use of water use. So you can see in the profile they're using. Neelam, one of the varieties I have developed here, is taking water slowly and making available water for seed filling better compared with this DICC 9073. So this is related to the root system. That is the pattern of water use and seed yield and a terminal drought in chickpea. And both papers are available, one in Journal of Experimental Botany and one in Frontiers in Plant Science. And just a pictorial way, you can see Neelam control plants in large bins, and this is the droughted one, and this is almost dead, whereas this is still continuing compared with the one. So it is the pattern of water use through the modification of the root system helped that plant to survive better under drought. The ICRISAT team, together with the people from Ethiopia, India, has used that information, which they developed, the Kasavagi et al., and now developed a number of releases. I think last year, India released a number of varieties with the drought resistance. And that work by Raji's group, they developed RILS 49580 is the large root system type. ICC 1882 is a small root system. And then they have got two other backgrounds. And what they call the QTLs for the root traits and various other traits in the LG4 location. And this is one of the examples where the physiology work led to genetic work using, combined with the molecular work, released commercial varieties of drought-tolerant chickpea in India. And I believe that that's also released or going to be released in Ethiopia. This slide may be a little bit shady, but basically I wanted to show that the structural and functional root traits identified as potentially relevant for drought resistant. So if you like, for example, root length, rice, wheat, maize, yes. Root biomass, yes. So there's a lot of now work showing that the importance of root system in relation to drought resistant breeding in different crops by different groups and so on. My last slide, Chairman, whatever we do, the strategy for strengthening the breeding process, because my topic is about breeding. I showed some physiology, then I showed how we can have efficient phenotyping system. I showed the physiological basis of whatever we found in water or nutrient uptake, particularly phosphorus. So the approach is like this: maximize the genetic diversity. We need to look at a large germplasm. Fortunately, the germplasm is available. Sequencing and genotyping of the germplasm collection. Phenotyping, I was focusing more on the phenotyping. Simulation and crop growth models, very important. Development of diagnostic markers, sequencing-based trait mapping, and gene discovery. That's good. In the past, people used to operate that, but we need to link with the improved genetic enhancement. Large number of crosses needs to be made, not just one or two RILs and NILs. Large segregating breeding population. Early generation screening is important because if you come later on, it's already fixed and you may throw some of the important traits. Genome-wide predictions, and of course, molecular-based selection and marker-assisted breeding, backcrossing and breeding. Rapid generation. Nowadays, we can, in our lab, even wheat and barley, one of our colleagues can take up to 8 generations per year. And in the legumes also we can take. So that is helping speeding up the process. And then genome editing is also happening. Operational efficiency in the breeding program, experimental design, a lot of development in biometrics, field book data, not just based on field books. Nowadays, all the pads and everything straight into the computer, analytical and decision support tools, use of databases in breeding program, and the more importantly, integrating with the breeding program. And that itself is not enough, because if you don't have the efficient seed system production and the agronomic practices and market access, it won't happen. So we need to strengthen the formal and informal seed system. And in India, it is definitely happening. faster varietal replacement rate. We can't really have 20, 30 years of varieties still sitting with the farmers, and we cannot really justify the research and breeding we are doing in a country. And timely availability of affordable seed. Some of these crops, seeds are very large, so farmers need large quantity, so seed production multiplication is important. Better soil health and optimized agronomic practices. Access of farmers to markets through digital agriculture is coming. We're just developing a paper on all this into Nature communication on that. So input from stakeholders, higher genetic gains, increased productivity, and integration with value chain and increased. And again, this review paper is available in Journal of Experimental Botany. So my conclusions, the last slide. Genotypic variability is the starting basis once you understand the basic physiology, and this provides a promising approach for selecting efficient root system for abiotic stress in this case, but also for biotic stress. Modification of root system architecture definitely could contribute to improvements in desirable agronomic traits. We have done indirectly, as I show here, Heron versus Gladius, but now we need to overcome that black box approach. So the recent developments, we were talking about all this in the past, but now it is happening. Phenotyping, imaging, modeling, molecular marker technology. Even country like India is full of wonderful laboratories in Delhi, Hyderabad, everywhere. And this will enable large-scale phenotyping of root-related traits and its use in breeding programs. And more importantly, we need to translate the physiological and genetic and even agronomic advances in understanding the role of root system in an improved adaptation to abiotic stress, particularly so when the climate change is already happening, and nutrient use, because we don't want to pump a lot of nutrients. It is costly, it's environmentally expensive, and this will lead to enhanced productivity of our crops for that, because we need to produce more per unit area, not expanding this. So we're talking about at least 50% more food by by another 50 years' time. Thank you again for the opportunity, Alamaru. Can I stop now?
Yes, sir, you can stop the screen
So
share.
I will stop sharing.
Rupali ma'am. Ma'am, unmute yourself. Yes.
Thank you, Professor Siddique,
Thank you.
for
Thank
your
you.
enlightening talk on breeding for efficient root system for water and nutrient uptake in crops. This is such a valued, beneficial, and significant information that you have passed to the students, researchers, and budding scientists to pursue a right research direction in grain science. You have certainly given us a perspective on what current research should be focused in crop plants to tackle biotic and abiotic stresses arising from climate change and dryland farming. Indeed, it is a great resource of knowledge coming from you on important root traits and root phenotyping to breed highly efficient crops with improved harvest index and breed yield. So thank you again, sir. I'm very happy to see great responses and some excellent questions on YouTube Plantgenomia channel. Professor Siddique, are you happy to take some questions from our webinar attendees?
No problem. I am happy.
Yeah. So here goes the first one. Is the elongation of the roots considerably a negative characteristic as it affects the distribution of the biomass, thus reducing production?
Yes, it's a very interesting question. So as I said earlier, for example, if you don't have sufficient water in the lower depths, or if your soil doesn't allow the roots to go deeper, then a deeper root system is not good. For example, you may have a soil in India, I'm talking now India, the top 30, 40, 60 centimeters is important. Beyond that, you have massive clay structure. No matter, even a jackhammer cannot penetrate, so you don't need to breed for that. So everything costs cost. For example, if you want to produce more leaf, it costs. If you want to produce more roots, it costs. So it's a trade-off. And that's why I showed the data that modern variety versus older variety. In the case of older varieties, there has been huge amount of root system and breeders unknowingly have moved that allocation into the shoot. So how much we can reduce the root system, that depends upon the environment. So yes, it will cost money, it will cost resources, but we need to optimize and develop idiotypes for the target environment.
Thank you for your response on this. The second question, to carry out research in root traits, what are the root traits we can look for the country with less resources and funds?
Well, a lot of those ones which I showed doesn't need a lot of resources. For example, you can grow things in PVC tubes and things. You can screen some of them in soils and go and dig through augers and things, which I used to do a lot. Many of them doesn't need a large amount of resources. You don't need a lot of equipments, but things like rhizo boxes you can fabricate locally. It's not very difficult in a country like India. PVC tubes you can get in India. In fact, we just did nearly 80 or 90 tubes in IRI, one of the PhD students. When it comes to genomics and other things, you have wonderful central laboratories in India, nationally based, etc. It's a matter of then, even in IRI, there it's a matter of collaborating and finding. First thing is that you need to see what is your research question. You don't need to jump into roots, and what are you trying to do? So read the literature and talk to your, so India is an ideal place to do a lot of research. For example, the early work done in Equisat, the Kasavagi et al. Work, completely done in the field. But a lot of the digging was done by backhoe, and then the laborers, the technicians and others went and collected the root system. And many of the techniques like dry weight, length, all those things doesn't need sophisticated equipments.
Thank you for your response on this. So our third question is in waterlogging condition, what root growth and architecture perform well?
Okay, so in fact, I'm just working on one of my PhD students' manuscript at the moment. She done some work on mung bean and black gram in your country, and mung bean is not very tolerant to waterlogging, and we have done a lot of work on waterlogging as well at UWA and my group. In this case, stages of waterlogging sensitivity is important. So you can have, for example, you plant the crop, the germination from the seed to up to a small, if there is waterlogging, you can study at that time. Then the seedling stage, you can study, and some species, the flowering is the most critical. So you need to look at the local environment. What is the likelihood of forming waterlogging? As Dr. Rao mentioned, you got the climate models, etc., and you can then define and say, now the climate is changing, torrential rain is coming at certain time, and it's creating waterlogging. So how does the waterlogging survive? One of them is the root system after decay, when the waterlogging is over, transient waterlogging, rapidly generating new roots. The other one is what they call the aerial roots. So they come from the node and they were able to breathe air. Then you can go and dissect the root and look at the parenchyma. For example, the one which is adapted will have an parenchyma, which then able to get the air in. So you can do large-scale screening, for example, in a field like in IRI and other places. You set up, then you grow them. At the right time, you have the flood irrigation. Make sure that you separate genotypes with different phenology at different blocks or something. From there, then you identify the one which is showing tolerance. Then you bring into the laboratory or in the glasshouse, screen house to understand what is the thing. So nodal roots, parenchyma, and escaping rapid generation of the roots after the decay. These are some of the ways in which, at least in the legumes, of course, in the case of rice, submergence rice, you know, a lot of work done by IRI, and now it's going on. So it's all how the plant is going to not be killed by ethylene production, and also the air, how can they get aeration through various processes. So there are physiological traits.
Okay. Thank you very much for the information. Now, due to time constraint, we'll take one last question. And the question is, can the hydroponic system you develop be used for fruit crop seedlings, for example, mango, guava, citrus?
Yeah, it is going to be a bit harder because these are all smaller pouch, but I'm sure that if you are looking for early stages of the seedling stage, one can adapt that. I'm sure that it can be done, but they are all perennial crops. So most of my work and our work has been to date in annual crops. It can be done, but in the case of mango and citrus, etc., it is possible to have field-based screening and then look at what is surviving and then take into the lab and have a look at it, because you can just plant them and then flood irrigation systems. The pouch system we developed potentially can be adapted, but not large trees, but for seedling stage.
Yeah, that certainly is a good approach. So thank you, Professor Kadambot Siddique, for your valuable insights on the question posted by our attendees. This is the end of the questionnaire session, and I now request Dr. Abhigna to take over the session.
Yes, before that, I would like to request Dr. C.H. Srinivasa Rao for his comments as well.
Thank you very much, Professor Siddique, for really enlightening us and with this, your scintillating lecture. I think all our youngsters must have benefited a lot, and in various aspects of genotypic variation and genome studies, breeding strategies, and also how you also suggested multidisciplinary approach for a efficient root system for abiotic stresses and biotic stresses, and how the integration of crop, water, nutrient production technologies are required for overall strategy. And for your for the students' information, I tell that recently in United Nations climate negotiations, these are the topics which have been discussed. A series of workshops are going on under United Nations UNFCCC global climate negotiations, where I take part as an Indian delegation to represent agriculture and loss and damages. Those sessions I follow, and very, very important. And also I remember Professor Yo Weissel from Tel Aviv University, who has written 16 books on roots. He always says that roots, the hidden off. So the hidden off has a major role in stressed environment. That is very, very important point, as future agriculture and food system will be will have to deal water crisis, will have to deal with nutrients, because Professor Siddique mentioned that phosphorus nutrition efficient root system. We don't have rock phosphate deposits in the world. We don't have, for example, India, we don't have potassium minerals. We are importing entire potassium. So that's why the topic is very, very relevant. And through these approaches, as he said, that 2030, we have to reach to sustainable development goals. And for India, especially South Asia, very, very importantly, SDGs, where large population, zero hunger, nutrition, health, and climate action. Thank you very much, Professor Siddique. And really, we are very thankful for your deep understanding and presentation, and a very, very in comprehensive way. Thank you very much.
Thank you.
Well, that was extensively informative. Thank you very much, Professor Siddique and Dr. Srinivasa Rao, for such a flawless progression of the knowledgeable session and for illuminating the subject to this extent. Thank you very much. Well, for the next event, I would like to call upon Dr. Sujeshri, Chief Operating Officer of Plantgenomia from Indian Agriculture Research Institute, New Delhi, for carrying forth the interview round.
Thank you, Abhigna. Many thanks for addressing our group, sir. So thank you both of you for the inspiring and informative insights delivered today. As we would like to know much about your vision and to get more suggestions, we have few questions, sir. So moving to the questions to Srinivas, sir. Sir, I would like to ask you, like for research capacity strengthening, there is a need to develop human resources. So we would like to hear from you the role and needfulness of research policies, capacity building, and training initiative to disseminate and implement science?
Thank you very much, Sujaya. Very, very important. In fact, we often in India, in many other countries, training is underestimated. And training, capacity building, and competency enhancement. These are the three key words. And whatever position is there, from farmers to young scientists to students to diplomats to everybody, training and capacity building is required. For example, I take the example of United Nations climate negotiations. Around 200 countries participated. We are only the 3 experts with a science background, agriculture background negotiate. But many are the lawmakers who negotiate agriculture. So it comes with the training and exposure. So that is the really matter. And if you see that other countries, you can see that they study agriculture but do medicine, medical research. Then somebody goes to space research. So it can, basic degree is one side, but with capacity building training, we can really move across all the discipline, which is really required. As far as NAARM is concerned, students are, especially during last 3-4 years, Sujaya, students are central part. We believe that students are the very, very critical. As Professor Siddique said, today's students, tomorrow's future of India or future of the world. So student training across the, in fact, all 74 universities, agri universities, we want to reach out. And we want to reach out to not only agriculture students, we are going to even +1 intermediate, 9th, 10th standard people to attract best talent towards agri education. Thank you.
Thank you, sir. Thank you for your thoughtful vision. And my second question is, sir, already you have given some insights to that point. To my next question, like, what is your key message for the student community on their skill development part to become very successful in their career?
There are, even while doing studies, there are many skill development trainings are there. For example, education technology. For example, I take many things. One, I take the example of NAARM, diploma in education technology. And while attending your own classes, you can attend these distance education courses. And another thing is that MOOC, massive open online class. Even while attending classes, while doing research also, you can still register some of the cases. Classes and research MOOCs are, we are training for teachers as well as learners. So learning also art, it's not easy. When we are making so much in the thing, learning and teaching is art. So there are many opportunities for the students, youngsters, and for training, capacity building. One is the distance digital education, MOOCs, and off campus, all other things. I think anybody is interested. In fact, you can put me email and you can refer NAARM website. So far this year, we have trained around 9,000 participants from the Forest, Indian Forest Office from India and Bhutan to civil servants to students. So around 9,000 we have trained each year, 8,000 to 9,000.
Thank you, sir. And sir, my last question is regarding our platform Plantgenomia. What are your comments and how you feel about our initiative in disseminating the science knowledge?
Certainly, as I said initially, actually, one good thing I learned, Professor Siddique, compared to all my earlier 26 years, 27 years of research in different Indian Institute of Soil Science, Bhopal, Indian Institute of Pulses Research, Kanpur, at IRI as a student, at CREDA, at different positions, and working with the students, interacting is a really, you always be really very, very energetic. Even you can gain the lost energy, you can regain. So that is one thing. And this platform is very, very important. You see, in a way, I am seeing you are all the leaders of future. And this platform, I believe that a leadership is inculcated. You yourself know, in fact, always our interactions, guides, seminars, symposiums are top-down approach. Here is the down-to-top approach. The students are managing everything. Recently, I attended last year, I was speaking Agricola in Tamil Nadu Agriculture University. Only BSc Agriculture students have organized everything, and they have made really critical components display system. So I believe that this platform is a great, and I personally from NAARM side, and anything is, even if you want to organize any physical, this kind of things, you are most welcome from NAARM. NAARM is the best infrastructure in India in all agriculture research education system, and we are always with you. Thank you.
Thank you so much for your supportive words, sir. It was very inspiring. Moving to Siddique, sir. Sir, we are grateful for the time you spent and the effort you took to share your thoughts and experience. So my first question is, sir, we know that you are a pioneer in your research area. So we would like to know like how you have started your career. So please tell us about your journey so far as an agriculture scientist.
Very interesting question. So as I mentioned, I was born in Kerala and did my high school in a government school there, then moved into a private college hostels. So you do your pre-degree those days, it's the +2. And as everyone wants to go for medicine, and I wanted to go to medicine, I don't know, that's what the ambition. And when I lost it for 6 marks, so if I had 6 more marks, it's very tough those days. There weren't many colleges, medical colleges. And then I joined for BSc botany or zoology, and I was not very happy. I just, within 2 weeks, I was just not interested because, you felt very sad. You didn't get what you wanted at a young age. But someone told me that agriculture is very good, and he was a cousin of mine. He was a block development officer, meaning he's an agriculture officer. Said there is one college only in Kerala, that's in Trivandrum. So I applied and got it there. It's quite a different environment. It's a professional college, 4 years, so all the students stay there. But I was not very keen. The first semester was psychology, sociology, extension, that kind of courses. So one of the professors, I do remember, he came and said, looks like you're not interested. If you're not interested, why you wasting your time? So that really challenged me. And then I just, so that first semester I had mostly B's and one subject I was C. So I had only one A. I can't remember how many units. Then I said, from there, I never looked back. I never got anything less than A. The A is the maximum you can get. It's a trimester system. Did very well. Then I was also very much into sports and things. A lot of friends. Then I learned about IRI because this place where you visit as part of your undergraduate program, All India Study Tour. Then these ICR exams. But the exams wasn't synchronized with the timing of Kerala graduation time. Anyway, I went and worked hard on that. And I got a letter in an envelope saying that you are selected for the ICR Junior Fellowship. You are asked to come for an interview to IRI. So I went there on a train. It's called a KK Express. It's a Kerala, Kashmir Express. There, it was a hot day. I remember Professor Rajat Day, Dr. Ravano. He's a tough man, but a very good interviewed. And then I said, I'm not going back. I'm just going to stay here. Then they said, oh, the results will be announced only after a couple of weeks. But anyway, they leaked out and said, oh, I got the first rank. And then that's it at IRI. But the whole thing is about agriculture. I won't go a lot. IRI then became the gold medalist, and again 4 out of 4, which I still keep it in my room. I think Rupali may have seen that 4 out of 4 is pretty hard in IRI because very, very competing. Only 4 or 5 students, and you also have PhD students taking some courses. soil science, good crop physiology, and very, very good teachers, Dr. Rajendra Prasad, Professor Day, then the water technologies, Professor Sinha, physiology, all those things. But then I got the admission for PhD, but I became very disillusioned because it's just what I'm going to learn. So then I joined for PhD as a gold medalist for 1 month. Then I got an offer from ICARA, International Center for Agriculture Research in Dry Areas, where it was just starting, and I went to Syria as the first. So my first job was overseas. I never stayed in India. I never voted in India. I left. My first salary was $1,100 US tax-free at that time. Can you imagine? So anyway, went there, and one very famous person called Dr. Mohan Saxena is now retired. He was the physiologist, agronomist. So he gave me whole heaps of work, challenging, and I did very well. Within 1 year, I became very unhappy again. I wanted to do my PhD, so I started applying. Finally ended up in University of Western Australia. And then changed my topic and learned a lot, worked with many people, farmers in particular of the state. I have now 14, 15 PhD students, and Rupali knows that I have time for students. Anytime they send something, I respond. But also I like to write things. Don't delay. You write, you learn, you read. Even for this particular seminar, how much time I spend, you can ask my wife. She says, you can go and give a talk, but I wanted to make sure that each and every slide, the color, the font, one to the other, will that slide be too much for you people sitting outside? If I find it is harder, then you will find it harder. So don't talk about things. Anyway, it's a long story. Then I ended up in Australia, but I do work a lot with India. If you look at the number of projects I had with India, Australia, India, Strategic Research Fund, the ICRISAT, started a master's program in Kerala Agriculture University on integrated masters in climate studies. Just recently finished a wonderful project with the Discovery Project. Can you read that? It's called Local Knowledge, Intellectual Property and Agriculture Innovation. This is based on a project which we had with Kerala, Indonesia, and then surveyed something like 400 to 500 farmers, rice, traditional rice cultivation, and a number of papers came out of that. So read, focus, ask questions, don't just repeat things which has been done by others, challenge, write, don't leave your data. If you do not publish, I tell my students that data does not exist, because it may exist in your computer, it may exist in your thesis. Thesis is easy, anyone can do it. Publish. And for example, Rupali's 4 papers is really her thesis. I mean, her 4th paper just came out yesterday, right? Yesterday, and so on. So that's what we had to do. Then bring a group of researchers together while supervising. Students go and talk to other students, not in the same division, not in genetics alone, not in post-harvest technology alone. Talk to other students. But also, leadership is not about the studies and publication, all other aspects. You just do it. So that's the story. It's a long story.
Thank you, sir. It's a big journey.
Yeah.
Okay, sir. My next question is, let me first congratulate you for being the highly cited researcher for Web of Science. Sir, well-deserved achievement. So with that background, please tell us your opinion in tackling the challenges faced by a student during the research and publishing their research outputs.
Okay. So first of all, you got only limited time. In our case, we have just say 3 years, 3.5 years, maximum 4 years. So your time is limited. You're not going to there to solve the whole world problem, number one. Because some PhD students think that I want to do this, I want to do that. Your research question, within the first 6 months of your PhD, within that time, if you cannot have the proposal done properly, then you are in trouble. So what happens is that that is the critical period where you have to listen, you have to read, you have to develop 3 research questions. Hypothesis you call, research questions. You must really have a critical review, very short script. What is the knowledge around that topic? The topic may come from your supervisor because a lot of times it's funded. but you need to have your personal signature. Then dissect into different compartments. As soon as you finish, keep writing. Write it as not as a paper, as a chapter. If you delay that writing, then what happens? You go into the next set of experiment. Then next set of experiment, then you come. Continue to read at night, because every day I'm struggling. I'm one of those guys. You can ask Rupali immediately. I send a paper, I send it to my students before they wake up, because knowledge is coming, new things are coming. And when you hit the paper to the journal, 85% of the papers you submit are rejected in a good journal. Some journals, 90% is rejected. Then you look at, you're writing for not you or your supervisor, you're writing for a peer group, very critique about that work, statistics, the methodology, repeatability, soundness, research question. So once you get that, then you can do. Why are you doing? Of course, after certain experiments, it will fail. It may not give the right thing. That's part of the learning. For example, many students, I had one student called Victoria from Chile. She wanted to do some work on brassinosteroids. I said, it's a waste of time. She didn't listen. So she wanted to do, I spent $12,000 of the work 1 year, nothing came out of that. Now I said, this won't work. Let us look at other aspect. So the second year she started, and then she produced 4 papers and gone. So, but in PhD, you don't have a lot of time. Read before you start and write down 3 to 4 research questions. And for example, Rupali's work was looking at herbicide tolerance in wheat. So she went and screened large genotypes. She developed an assay, published that work. From there, she found the variation. Then she started crossing, made the RILs or NILs. From there, we wanted to know the genetic basis and the physiology, and then RNA type of approach. Well, when she started, probably she didn't know she will do that. But in that case, the right combination of supervisors and her own initiative, it helped. But read, write, then question. Is it new? What is new about it? Why should I do? Why should I repeat it? Go into the library at IRI. You will find the same bloody thesis, sorry to use that language, is repeated again and again. You change one treatment, the other, and then a thesis is produced. No one read other than your supervisor, you, and examiner. After PhD thesis, I don't even look at that thesis. So if you publish, world knows about it.
Okay, sir. Thank you for your thoughtful advices, sir. And my next question is, with the background details you have given on regarding the University of Western Australia, it has inspired us to pursue our goals. So many of our dedicated students and passionate researchers are part of this event today, who are very much interested to pursue their higher studies in the abroad universities. So, sir, what sort of tips you can give them to attain the target?
Very, very, very good question and very easy to answer. In fact, I can give a lecture on that, but I'll make it very brief. Number one is that you wanted to go overseas, like Professor Srinivasa went to Tel Aviv and he learned a lot from there. Why do you want to go to overseas? That's number question one. When you are doing your master's or whatever, you need to look at who are the people in that area working globally in a very area. So half a dozen. Then you look at their papers, etc., and start learning. Is there anything new you're going to learn? There are different ways of getting. One is that scholarship is very, very competitive. So in other words, to get the scholarship, so let me say one example. About 3,000 to 6,000 students apply for a scholarship at the University of Western Australia. I used to be on the scholarship committee. This is across medicine, engineering, agriculture, humanities, arts. Of the 3,000 to 6,000, many of them drop back. Either they don't have the adequate language skills, IELTS or TOEFL scores. Second, they don't know what exactly they're doing. They send a little CV and say, I want to do PhD, but then many of them won't respond. You need to write certain things about yourself, why you want to do it, what is the area you want. To draw the attention of those professors who are very busy, like Sunday today, I got a lot of other things to do. So normal professors will just look at that and say, it's full of spelling mistakes, it's not good, he hasn't even thought about it, so then they just ditch it. So you need to impress that. The most important thing is that your masters, you must publish one paper at least these days, because We can't just tell, oh, your thesis, we don't have time to read your thesis. And how do you assess your ability when you contact to me or anyone else? So publish that paper in a good journal, preferably as a first author, that is good. Do masters from a reputable university in your own country, institution. Many universities are not even known. I'm not saying all Indian universities are bad. That's not true. Even, for example, Kerala Agricultural University, someone applied, they don't know. It's a good university, I know. I was a student there. Even an Indian Agriculture Research Institute, for example, they don't know because it's not a university. It doesn't come under university. So government of India has to do something. For example, when Rupali applied, she had, and first time applied, I can tell that now openly, Rupali, the ranking committee just said that that college, that university is not a good university. So I leaked it out because I think you came and talked to me. Then I challenged the scholarship committee. Then second round, I just wrote a paragraph about it. Because I was there, they took seriously. There's another candidate at the moment with me. He's doing PhD, Mukesh Chowdhury. He's from IRI, from genetics division, but he's a scientist in Wheat Research Center in Ludhiana, ICAR Wheat Research Center. So when he applied, the other supervisor, I was not here. They simply said the IRI, he hasn't done a thesis. What is his IRI and all that? So I got really upset about it. I wrote a letter to the scholarship committee. Next time you ask if you don't know. Anyway, he got. So I am not going to be there when they apply to another university or something. So good place, good masters, a good paper if they can. If without paper it becomes difficult to get a scholarship, then you don't blindly go and say, sir, I want to do a PhD with you, because that person may be an animal scientist, but you want to do plant scientist. So you have to do your homework. What exactly? Scholarships are extremely difficult. So I got 3 students coming from India at the moment. One is from IRI, one sitting in Hyderabad, and another one. They're all waiting for the border to open, but it takes up to sometimes 2 years to get a scholarship. And there's many universities, US, UK, Canada, Australia, New Zealand, Germany, and so on. So if you fail at one place, don't worry, try other places. But it needs a lot of time and effort.
Okay, sir. My next question is, sir, finally, what would you advise the young researchers who are passionate about their agricultural research? And also, I would like to get some comments on our Plantgenomia activities and initiatives, like in disseminating this kind of information.
So I'll first tell about your initiative. So as I said, I didn't know anything about this initiative. Once I had an email from Rupali and also someone else, and then I saw various times. I think this is a great initiative. Can you imagine, without spending any money, for me to fly from here to India, giving this talk without any interruption, well organized. Alamaru was in particular communicating with my secretary, and she's very, very careful, and she asked questions. She want this, she want that. But it was all provided and then worked out at time. It's fantastic. It's even for big societies and others, they cannot organize this well. And no interruptions, very little time wasting. You got into the thing and you chosen the topic, and you got, I don't know how many people attended, but one lecture attended by this many people, I'm sure that even if 5 or 10 learn something from this lecture, is an enormous thing. So it's a great initiative. This is what I call leadership by young people without any other support or anything, by your own initiative, sitting in different institutions. This is something which I like. You know, you are in IRI, and Abhigna is in Hyderabad, right?
Yes, sir.
Yeah, and Almaru is sitting in somewhere in Punjab, right? At the moment. Yeah,
yes,
Punjab.
yes, lovely
so,
professional.
Look at that, then other people are there. So it is incredibly good, because India is a lot of silo mentality. Delhi University doesn't talk to JNU. JNU doesn't talk to IRI. I'm giving a general picture, although there are exceptions. And as I said earlier, division genetics doesn't talk to division physiology. Division physiology doesn't talk to agronomy. What is this about? It's about agriculture. So this is excellent. Keep going. Get some more people internationally, and then don't spread too thinly, but focus quality lectures, which is relevant to that. That's good. Now, hopefully, when the COVID situation improves, You go and look at some big conferences happening in India. You go and then develop a workshop around that. Tell the organizers, we wanted to develop a small workshop. Then the dignitaries and others coming both locally and internationally, you bring them and form a workshop. So expose. It's good for you, good for the country, good for everyone. Now, the second part of the question was? What was the second?
Yes, sir. It was regarding like the kind of advice you can give to the student community.
Yeah, I think the main thing is that there's a lot of opportunities, full of opportunities. India is really moving, so as other countries. Let's take India itself. Agriculture will be forever there. Let's not even call us agriculture. Food industry is the largest industry in the world. You can challenge me. You can say, oh, no, sir, IT industry is important, medical industry. No, the food industry is the largest industry in the world. That is the production, transportation, the processing, or supermarket chain, health benefit, all this. I just published a paper in Nature Plants. If you're interested, go and read about the hunger and malnutrition. So the whole country, every country, food is the most important. COVID-19 has really showed that the importance of the food system and the food quality, the food and nutrition, the food traceability, the production system, environmental issues, the global challenges, the climate change, and so on. As years come, the food and health become the key thing. The countries will be interested in the cardiovascular, non-communicable disease, motor neuron degenerative disease. All those is linked with your food system. Microbiology, the soil health. So huge area, exciting opportunities. What you have to do is that focus, do the best. You may not be the best in everything, but you have certain area and try to work, try to broaden your horizon. These days, when we were students, Dr. Rao, to get a paper, you have to wait. You write to a professor, they send it by email. You have to wait for 3 months, 4 months. Now, on the tip, you can go and say, if I said a paper, you go and look at it, you will find it. If you can't get it because of the library, you write, and I'm sure that the professors will send it immediately to you. It's an exciting opportunity. Agriculture is going to be the next exciting thing, and all other technologies. And in fact, IT, the whole thing is going to come to agriculture. Sensors, data, artificial intelligence, robotics, but you still need people with the brain. All these things, you can have the toys. If you got all the genomics, etc., if you don't know how to interpret that data, what is it about? You know, I give a talk. If I don't know the soil science, if I don't know the plant science, if I don't know the fundamental thing, I cannot give a talk like that. I can look at the data, study, but I won't be able to give a talk. So broaden the horizon, focus, the best you can achieve, and say, I am the best, and then try. And you need not to compare with other students. So, and read. I mean, at the night, I don't get someone coming and giving the tablets to me. I had to read, and the piles of papers coming out every day. But then you have to be careful what you want to read. Don't believe everything written in the paper either. You question and say that Professor Siddique is wrong in that. And then you write in your paper, although they did this work, there was an error. And that will be then quoted by people. Hindsight, I never chase for hindsight. The first thing is that publish. If you don't publish, you won't get citation. So 50 to 60% of the paper, some of people have published, don't get even one cite, meaning it is useless. That means it's not read by, what is the purpose of publication? The purpose of the publication is to let others know what is going on. They take it from there, the knowledge build up. So you need to be novel in that. And hindsights and things will come. If you don't publish, there's no. And you have to pump it up. You have to publish as soon as your research is finished. Some are called perfectionists. They will delay for another year. By another year, it is gone. Don't delay. You will never finish everything. Research is called research. It is existing somewhere. You're going and researching. It's not you made it. It is there. And you can research it. Then you tell the world, this is what you found.
Thank you, sir. That's the end of the question answer session. And we have greatly enjoyed the talk. And thank you for sharing new insights in achieving our goals to both Srinivasa sir and Siddique sir. Thank you, sir.
Thank you, everyone, and have a good Sunday, rest of the Sunday, and we'll see you sometimes. Thanks again.
Yes, Abhigna.
Yeah.
Truly an amazing interaction to have waited upon. Thank you, Dr. Sujeshri, for executing the interview that most certainly was quite intriguing. Now, wasn't that a mind-blowing session we had? So thanks a lot. You guys have been a brilliant audience. And I would also like to acknowledge the presence of the chief guest, Dr. C.H. Srinivasa Rao, and the speaker today, Professor Siddique, of course. Subject specialist, Dr. Rupali, Chief Operating Officer of Plantgenomia, Dr. Sujeshri, founder of Plantgenomia, Mr. Alamuru Krishna Chaitanya, and all the efforts put forth by my fellow mates, A. Sai Teja, K. Sai Timmarao, K. Sai Sumant, Srimoy Bhattacharya, Aman Tutlani, P. Sriraj, V. Anila Sweta, G. Vinay, T. Bhavyashri, and T. Sai Reddy, who contributed to make this event a success. Reaching the end of this, I would like to clarify the process of attaining this certification for the same. A feedback and a certification link has been shared in the chat box and the YouTube comment box as well. You are all kindly requested to go on to the same, fill it up, register yourselves, and thereafter, within a slot of 1 week, your certificates will be shared accordingly. And if there is some error and you do not receive your certificates after a week's time, kindly visit our official website www.plantgenomia.com. Please note that the link is valid for only an hour. I deem it a great honor and privilege to propose the vote of thanks on this memorable occasion. First and foremost, I would like to thank our chief guest, Dr. C.H. Srinivasa Rao, who took his time to grace the occasion, along with our speaker, Professor Siddique, whose tremendous efforts left us with an enduring knowledge. I would also like to thank the course support and course subject teams, Dr. Rupali, Dr. Sujeshri, and our remarkable audience for contributing to make this event a huge success. Thank you, everyone.
Okay, all the best again.
Thank you, everyone. Thank you, sir. Thank you very much. Thank you.
Okay, so I can leave now.
Yes.
Bye-bye.
Thank you very
Bye-bye.
much.
Bye, sir.